Yeah, turn on the recording. So 19 people have joined. Should we start? Because we have a lot of ground to cover. First of all, we missed 2 sessions, and then I have added 1 lecture. So
Yes, sir.
we have a lot of ground to cover. So vis-a-vis. So okay, fine. Please bring out questions if you have any. So nobody has any question?
Good evening, sir. Sir, I have a question.
Yeah.
Sir, you mentioned in the lecture that for next gen sequencing, we can simultaneously perform millions of reaction and sequence like a large number of copies of gene. But for Sanger sequencing, in a single reaction tube, only like one specific gene can be sequenced. Like if we mix 2 templates, then like we won't get the appropriate sequencing. But
Yeah.
so my question is like, if we add a primer for the gene to be sequenced, specific to the gene to be sequenced, then in Sanger sequencing, even if we have 2 separate templates, then why would there be a problem?
Yeah, very good question. Okay, so these are the reasons why we need to think from a very elementary perspective, like forget textbook and think. So let us think how Sanger sequencing is read. Okay, so you imagine that you have a, okay, let us try it. Of course, I do not have my stylus, but still let me try. Okay, so imagine this is a tube, okay? Now here you have DNA number 1 and here you have DNA number 2. Okay?
Okay.
Now you will be adding the primer for both of these. Here primer 1 and here primer 2. Okay? Now how will it work? That as soon as it incorporates a fluorescently tagged DNTP, the reaction will stop, and the camera, the microscope, will detect the fluorescence, right? And based on the color, it will know whether the last base incorporated was an A or a T or a C or a G, correct?
Yes, sir.
Yes, sir.
Okay. Now let us imagine that you can see my thing, right?
Yes.
Yes, sir.
Okay. Now let us imagine that this is 5 prime, 5 prime, this is 3 prime, this is 5 prime, this is 3 prime. Okay. So you will have a primer over here and a primer over here, right? So now this one, after the primer, the first base in the top one, after the primer, the first base is a C, right?
Yes, sir.
So when it will incorporate a G, it will stop. And the lower one, after the primer, the first base is a T. When it will incorporate an A, it will stop, right?
Yes.
Now, your fluorescent camera, microscope, all it knows is that there was one green incorporation and one red incorporation, right? How, first of all, how would it know which one was incorporated in which one, whether it went in this one or this one? Do you realize what I'm saying?
Yes, sir.
Yes, sir.
So what happens is that you actually get this kind of overlapping peaks. You get this kind of overlapping peaks. As a result, you will not be able to assign sequence of either one. Is it clear?
Yes, sir.
Yes.
Okay. Next question.
Am I audible?
Yes, you are audible.
Hello. Yes. Sequencing method. So actually, I couldn't tell the algorithm part. I mean, how algorithm can be used and how.
Adil, you are not very clearly
Yes,
audible.
sir.
Now it is better. Please go again.
So actually, I couldn't understand
Hello?
the whole genome shotgun sequencing method. It is basically the hello. Hello.
You did not understand the shotgun sequencing method.
Yes. Sir.
Okay, that's not a problem. Adil, can you type the question in the chat box while I take some other question? And
Yes, sir.
then
Yes.
I will
Yes,
respond to your question properly.
Yes, sir.
Sure.
Sure. Sir.
Anybody else?
Sure, sir.
In the meantime, anyone else would want to ask any question?
Yes, sir.
Yeah.
Yes, sir. Okay.
Sir, I have a question regarding topo cloning. So in topo cloning, I learned that the topoisomerase, but it will bind to two strands, but it will only cut one strand.
Yeah.
When topo cloning is done, so two topoisomerases join at two places on two strands. So topoisomerase will recognize CCC T sequence. So if one strand CCC T sequence is at that place and at one place, but is it possible that in the lower strand, just below there, just the near place there is a CCT sequence? I think if in the lower strand CCCT sequence is not just near, it is very far, so it will not be able to cut. So that's my question.
I did not understand the question, Ubed. So, just to give you some idea, that the topo cloning pretty much works exactly how topoisomerase works naturally. So when DNA synthesis is happening on a distal part of the replication fork, DNA gets continuously twisted around it itself, right?
Yes, sir.
And then topoisomerase cleaves it so that it unwinds, and then there is a ligase that joins it back again.
Yes,
right?
Sir.
And
Yes, sir.
there is basically a mechanical switch that dictates when it will be cleaved. So
Yes,
if your question is about the sequence specificity of topoisomerase,
yes, sir.
then you have to understand that this is a commercial product. So the company has worked extensively to ensure that in the plasmid, there is no non-specific action on topoisomerase.
Yes, sir. Sir, actually, it cuts only one strand, one topoisomerase. So they have joined two topoisomerases.
Yeah.
Sir, my question is that both two isomerases, they will join at the same place, above and below. For that, they should have the above and below should be the sequence of the topoisomerase.
Well, I think it is a trade secret. It is possible that they have taken topoisomerase from two different species which have slightly differing sequence space. But honestly speaking, I do not know the molecular detail of how topoisomerase works.
Okay,
Okay,
okay, sir.
so I would not hazard a guess.
Yes,
I
sir.
really do not know the exact detail.
Yes, sir. Sir. Another thing, when I was reading topocloning,
Uh-huh.
it was written that the tag polymerase has a property that it adds one nucleotide, A,
Yeah,
adenine,
yeah, yeah,
in the
yeah,
non-template fashion.
yeah.
Sir, why it adds only adenine? Why not all other? And what is?
I do not know that. It is a property that has been characterized, but that's all I know.
Sir,
That why A and why not some other nucleotide, I do not know that. But
okay.
you should know this, and I am very happy to tell you this, that Invitrogen is a company that was born in front of me. So somewhere around third or fourth year of my PhD, Invitrogen representatives came to my lab, and I was their beta tester for their TA cloning vector. Like, I am sure I'm not the only one. I was
Yes,
one of
sir.
the beta testers for their TA cloning vector. And it completely changed the way we were doing cloning in those days. And I knew that this company is going to go far. And today it's a behemoth. Okay.
Yes, sir.
This company is hardly 20 years old.
Yes, sir.
Okay. Okay, so now Adil has typed his question. So let me read that question. Okay, Adil. So I think that you got it slightly wrong. Let me explain it to you how it is actually done. So Adil's question is that how is shotgun sequencing done and the sequence assembled? Okay. Um, let us do a hypothetical thing. Let us imagine that this is the actual DNA. Okay. Now, when you fragment it using a restriction enzyme or limited sonication or whatever, you will make multiple fragments. Okay. There is it is because these are all non-specific thing. So the fragments will not be clean. There will be a lot of overlapping fragments. Is that understood, Adil?
Yes. Uh,
Okay. So now each of these fragments will then be cloned in a plasmid. Okay. So that is your nowadays you don't even need to clone it in a plasmid. You can actually, like when Craig Venter did it, he did it like this. So shotgun sequencing was the concept was promoted by Craig Venter. While Francis Collins's group at NIH, they were sequencing from one end, and then they will be reading the sequence here, and then they will design new primers from here, and then they will sequence here. And so it was becoming a whole lot of problem. So what Craig Venter did is he simply fragmented the DNA and cloned it in plasmids and then sequenced it. Okay. And then used, very high powered computer like software to do mega alignment. So you can imagine that if you sequence all of these, then there is a possibility that because I cannot do it in a better way, I will do it like this. That you know that there will be a overlap between this and this and this, right? So that will create what is referred to as a contig
Yes,
from
sir.
here to here. Okay. Now this contig will have overlap with this
Yes,
and this. So the contig will be extended. So this is how the whole assembly is generated. Does it make sense? Adil?
yes, sir.
So,
And so I want to say, sir, how for that the valuable knowledge is for that.
Adil, I cannot at all understand what you were saying. Why don't you try something new? Like if you have a phone number of any of your friends, why don't you call that number so that I can hear your audio voice from the phone? Because I cannot hear your question at all. Adil?
So should I type in again?
Yeah, you can type it. You can type it. Yeah. Someone else was
Thank you,
trying
sir.
to.
so,
हाँ आकांक्षा बोलो.
so, so, like in this massive alignment, like you're telling where the contigs are extended. So, like there is also a scope of error here, right? Like maybe the fragments, like the overlapping things that we get, maybe we can find like it in other part of the genome also.
That is extremely unlikely, Akanksha. Like, of course, there are some repeat elements in chromosomes. So those will be taken care of by the algorithm only. Typical repeat elements that would bother scientists would be the telomerase region. But if you ignore that, then the possibility of a stretch of 30 nucleotides or more being identical to each other is extremely low, because the probability is 1 by 30 to the power 4, right? 4 to the power 30, right?
Yeah.
So that was not a problem. What did happen though, that certain parts of the genome remained uncovered, like so it was a humongous see, this method will work if eventually you can form a contig for the whole chromosome, right? If you have a
Yes.
contig like this and a contig like this, you have no way of knowing whether they belong to the same chromosome or not. So assigning them to individual chromosomes took a whole lot longer.
Okay.
Okay, Adil, you have posted your question, right? Okay. Oh, you are asking how we can design algorithm for that, algorithm for the alignment.
Yes, sir.
Okay, so I am the last
Yes,
person
sir.
to know that, I barely know how a computer looks like. Okay, so for that you have to ask Professor Hamim Zafar or Professor Shankar Ramakrishnan. I have zero idea about algorithms. Okay, someone else raised hand.
Okay, sir.
Who is it?
So I had a question.
Pucho, Pramit?
Sir, in the first slide in this presentation itself, so regarding the Shreya's answer, like your answer to Shreya's question. So we have two different templates and primers to both the different templates. Is it just for, answering the question, or I just missed something, or I misunderstood?
Yeah, like I was explaining to Shreya why in using Sanger sequencing method, why we cannot sequence more than one DNA at a time.
Okay.
So Sanger method dictated that every single piece of DNA will have to be pure and then only Sanger method can be used on each one of them individually. While next generation sequencing does not require you to purify individual pieces of DNA. Like they are spatially separated even though being in the same reaction mix.
Yes.
Okay, Akanksha, you have another question?
Yes, sir. So it's related to cDNA synthesis. So like in the slide, it was showing that after the cDNA is synthesized, there's a poly G sequence that is added to the 5' end of it. So
Yeah.
my question was like, how do we add that to a single strand? And like if we put ligase, then the other strands will also join together. So
No. So if you look at the slide carefully, then first the RNA is digested with alkali, right?
yes, sir.
So now you have only one strand. You have the in our language what we call is the first strand cDNA. Can you see my rough slide?
No, sir.
No, right? Okay. This is a bad feature of Zoom. Okay, so if this is your RNA, see, I cannot do the detailing, oligo, poly A tail, etc. Unfortunately today. Then when you make this first strand, like the complementary of the RNA sequence is made, right? That is the DNA. So this yellow color one is the DNA.
Yes.
This is referred to as the first strand cDNA. Okay, like okay. So this first strand cDNA will be made for almost all the mRNAs in the milieu, right?
Yes.
Now you digest the RNA with alkali.
Yes.
Okay, so RNA is gone. Now you put your poly G here, and you will be doing that for every first strand cDNA.
So like how is this added there? Like what's the enzyme or what is the
It's a
process?
ligase. It's a ligase. It's
Okay.
it's a very specialized ligase that only works on for single strand DNA.
Okay, all right. That was my question. Like if there is something specific.
Yeah. Okay, Ayush, you have a question? Please go ahead. Ayush. Okay, Navneet, you go ahead then.
Yes, sir. Can you hear me?
Yeah.
Yeah. So my question is related to Sanger sequencing. So you mentioned like if there are two templates, there might be chances that there are two nucleotides at a particular position, so that you get two peaks. So what if even with the same template, what if the wrong nucleotide gets incorporated? In that case, do we know the nucleotide at that particular position, or how do we proceed from there?
I did not understand your question. That are you saying that what if a wrong nucleotide gets in?
Yes, sir. What
Well,
if we get two peaks?
huh?
And so I was wondering whether how we can know the correct nucleotide in that position.
Okay, so this is not a theoretical problem. This is actually a practical problem. That
Yeah.
Everything said and done, things don't work exactly as you read in the textbook. Okay, so it does happen, but what you need to understand is that when you are doing Sanger sequencing, you are not sequencing one molecule of DNA. You are sequencing thousands of the same molecule of DNA. Okay, and because you are using a primer, so say, let us make it up. Say this is the DNA, and this is the location where the misreading will happen. Okay.
Yeah.
So if this is your primer, so from the primer, the distance from the last base of the primer to where the misincorporation is the same, right?
Yeah.
No matter how many different molecules is being sequenced, right?
Yeah.
So now there are two ways things happen. That majority will incorporate the right one, some will incorporate the wrong one. So that will be seen as this. Okay, so you will hazard a guess that the larger peak perhaps indicates the right base. But sometimes both the peaks will have same intensity, and you will have to do the sequencing again and hope that it will work better next time.
Okay. So in this case, do we run like different tubes? I mean, the same template with in different tubes so that we can at least make a
No,
correct
see,
guess.
we do all sorts of things. Sometimes what we do is we design a primer here and then try to read the sequence from this direction. And that very often resolves the problem.
So in the same tube, we can add a different primer.
No. A new tube. See, in the same tube, you cannot have two elongation reactions going on.
Yeah. Okay. Okay.
Hello.
Yeah. Got it.
Yeah.
Am I audible now?
Yes, Ayush, you are audible now.
Apologies for the last time. I guess there was some network issue.
No problem.
So I have few basic questions from the lecture that you posted. One is why, as you mentioned in the lecture, why cannot we do RNA in situ hybridization for C. elegans?
Yeah, like I do not know. I think their RNases are so two problems. One is that the tissue density is very high. Like if you ask me, because I work with both mouse and chick, chick is the best system for doing RNA in situ hybridization. And of course Drosophila. But C. elegans, I guess, has a very dark background, so the contrast does not come up. It is nothing to do with molecular biology, it is to do with the practical problems.
Okay.
So, but C. elegans is more of transparent, no? The C.
Yeah, but
elegans, we can
there
see
is some problem with it. You cannot do RNA in situ hybridization on C. elegans. And it has nothing to do with the molecular biology part of it. These are very specific things. Only a C. elegans biologist will know.
Okay. And another question from RNA in situ hybridization itself. So in one of the lectures, you mentioned that to revalidate the data, like if we are studying some developmental problem, to revalidate it, we usually prefer RNA in situ hybridization rather than qRT-PCR.
Yeah.
But like if we have a problem like in a tumor tissue, we are checking for a condition where, I mean, how gene expression takes place in a tumor tissue as compared to a normal tissue. So how do we revalidate it since we cannot do RNA in situ hybridization?
Yeah. So see, like by definition, the tumor tissue is a homogeneous population, like almost a homogeneous population. Okay. So there it is. So let me first elaborate that why in developmental biology context, RNA in situ hybridization is very, very important. So you know about kidney, right?
Yes, sir.
Do you know how many different cell types are there in kidney?
So there are nephrocytes and
there are 80 different cell types in kidney. Okay. Now, the fact that there are different cell types automatically means they must have slightly different molecular biology, right?
Yes, sir.
So now, if you do a RT-PCR, how would you know which cell you are analyzing?
That is actually an issue because when we isolate the RNA, it will come from all the neighboring cells
Yes,
as well.
yes. So that's where RNA in situ hybridization does a spectacularly good job. But as you rightly said, that if it is a cancerous tissue, your question is actually very different. In the cancer, when you are trying to look for an mRNA, you are typically trying to understand that what kind of genes are being expressed in the cancerous tissue so that you can grade the cancer, or you can very specifically identify the type of cancer. So there, the cell type specific expression is less of an issue. The collective behavior is more of a like a objective, right?
Okay, sir. And
Yeah.
another question. So, in the EST analysis, as you said that in one of the steps we have to grow the bacteria on 96 well plates, right? In each of the plates we are growing bacteria. But I have a very practical question. So I have worked with bacteria a little bit. So a simple or the basic requirement for bacteria is it is grown in large conical flasks so that there is proper aeration as well as the amount of nutrients available for the growth is also high. So if we are ultimately doing plasmid extraction and sequencing, if we grow them in 96 well plates, then the yield must be low. So how do we compensate for that?
Okay, so let me see. Okay, so like I have done some 700 of this. So let me show you what I used. Do you see these plates? Oops. Stupid site. Okay, can you see my screen?
Yes, sir.
Yes, sir.
Okay, so this is how a 96 well bacterial culture plate looks like. Okay.
Okay.
And it will have this breathable membrane on top of it. And you actually, there is not much utility of shaking it because not much swirling can be achieved. But like I have done it, and we would still get more than couple of micrograms DNA per well, and it was all like I was the robot in there in that case, and I used to use this. Okay, let me show you that as well. Okay, can you still see my screen?
Yes, sir.
So you see these pins?
Yes, sir.
Are you still
Yes,
seeing
sir.
the bacterial plate?
Yes, sir.
What, yes, sir? Are you seeing the bacterial
Yes,
plate?
sir.
No, sir.
No, sir. No, sir. Pins.
Pins we are.
So this is how it is done. That you will culture culture the bacteria, and then you will do the mini prep. There are 96 well mini prep format is there from most of the vendors. You will isolate the DNA, and then you will print those on the matrices, and you will actually take from the culture and inoculate a new one using these 96 well pins, so that you do not make any mistake about the location of a particular culture. And this time you grow them and spin them and freeze them and keep it in the minus 80, so that after you have like there is a reason for you to be interested in a particular spot on your array. From array you go back to the specific 96 well plate, and from there you go to the specific well to get out the DNA and sequence it. Okay.
Okay, sir. And one more question, last question. It is in one of the slides you mentioned that on the microarray slide, there are probes that are spotted by technology similar to DNA synthesis, right?
Yeah.
So my
not
question
probes,
is,
those are not probes, those are the templates.
Right.
Probe is what you add afterwards.
Right. Apologies. So my question is, since you also mentioned that DNA polymerase cannot synthesize de novo, right? And
yes.
on the slides, there are no templates available, as well as no primer is also added. I suppose I don't know.
This is chemical synthesis of DNA. Sanger also, who is it? Not Sanger. Who? Sorry, I'm forgetting.
Maxim
Gilbert.
Sir.
Maxim Gilbert.
Maxim Gilbert.
It is chemical synthesis of DNA, like where you use this FMOC to block one, so that you extend only in one direction.
Okay.
So that's what is done. Okay, Navneet has some questions.
Okay, I misunderstood it.
Okay, yeah. Navneet, you please go ahead.
Yes, sir. I have a question on EST. So, while preparing EST, why is it that they are not sure if the sequence of the mRNA is complete? Like why don't we get the complete gene sequence?
Yeah, it is not the
similar
sequence.
to cDNA library.
It is not the sequence. It is the copying of the mRNA is not complete.
Why is it so, sir?
Okay. So because the mRNA or RNA, it is single-stranded, right?
Yeah.
So because it is single-stranded, if there are stretches within the same RNA that are complementary to each other, that they can form double-strand, right?
Yeah.
Your tRNA is the best example. tRNA has very defined three-dimensional structure, right?
Yes, sir. Yes, sir.
That is because of intramolecular hydrogen bonding, right? Or base pairing.
Yes.
Yes, sir.
So sometimes this intramolecular base pairing will be so strong that the reverse transcriptase will not be able to melt it.
Okay.
Now, the other problem with the reverse transcription is that the reverse transcriptase optimal temperature is 42 degrees centigrade. Okay, that is not a high enough temperature to melt the secondary structure. Like while your PCR polymerases, TAC or PFU, they operate at 72 degrees centigrade, right? That is very good to melt any secondary structure. So here is your business idea. If you can make a temperature resistant reverse transcriptase, you are looking at a few $1 million.
Okay. So
yes.
What I get from previous discussion also that it's the same problem with the cDNA library preparation as well.
Yeah, because cDNA library preparation is dependent on successful complete synthesis of the first strand.
So at the time of preparing ESTs, these high temperature RT enzymes were not available, is it?
Yeah, no, it is still not available. The best we have is, I think, that works at 50 degree.
Okay.
That is still not good enough.
Okay.
Sandar?
Yes, sir. So, I had a question. I have a question regarding the de novo pathway. Like in the lecture, you said that if I'm not mistaken, that DNA synthesis cannot be done. Do not occur through de novo pathway, but RNA does. So,
Yeah.
what is the reason? Like why RNA can be
Well,
synthesized with de novo, but DNA can't?
okay, so I will not be able to tell you the structural basis of this. It is just that, it's a fact that RNA polymerases can synthesize de novo while DNA polymerases cannot synthesize de novo. I just know it as a fact. But what is the structural basis of this fact, I do not know.
Okay, so there's one more little doubt. Like in the iron torrent
Uh-huh.
sequencing, like there are wells containing same copy. I mean copies of same DNA, right?
Yeah.
And I mean one of the nucleotide A, T, G, or C is flooded in that well. So
Uh-huh.
is there any chance that some of the genes do not get that nucleotide? Like if suppose A is flooded and A has to go, like there is some T, the next one is C, where A will go and bind. But is there any chance that some of the DNA will not get the A, like even if there is C present there?
I did not understand your question clearly. Are you saying that?
Sir, I'm saying,
Huh? Go
sir,
ahead.
I'm saying ki, like there are wells. So there are wells now, different wells, and in each well there is multiple copies of single DNA, right?
Mm-hmm. Mm-hmm.
Now we flood A, T, or G. I mean A, T, G, or C, any of the nucleotides in each of the
Mm-hmm.
wells. Suppose, okay,
Mm-hmm.
and suppose A has to go there, like the next sequence is there T, like T is already present in the template, and A has to bind with it.
Uh-huh.
So is there any chance that there are multiple copies? So is there any chance that some of the DNA will not be able to, I mean, by chance, the A do not incorporate in some of the DNAs? Is there any chance? Like
Um,
my actual
well,
question is,
I understand what you were saying. That
okay,
like if that happens, then you might eventually read wrong sequence in the same
right.
spot. You might get multiple sequence variations, right?
Yeah, right. Multiple sequences, yeah.
Yeah.
So
So
if there's multiple sequence, then
yeah,
yeah,
I am
please proceed.
reasonably certain that that happens because see, most of the biological processes, the way we do it in vitro, they are not 100% efficient. Okay, so I'm sure there are some mistakes like that, and I I do not know. Okay, but I want to believe that there are algorithmic measures to take care of those. Okay, like
Okay, yeah,
statistical measures to take care of those.
okay, okay. Thank you so much.
Akanksha, you have another question?
Yes, so like in the last class, you asked us to like go through the URFs and all.
Mm-hmm.
So what I wanted to ask, like the one gene that we took into consideration that had a start codon and a stop codon.
Mm-hmm.
Uh, but like what I wanted to ask is there was one gene I came across. I still have to go through it again. So it was OASL. I do not remember one or two, but in that what had happened is it had a start codon upstream,
Mm-hmm.
but it had no stop codon. So like what could happen in this case? Like we'll have an different peptide altogether.
Did you see whether the reading frame is the same or alternative reading frames?
Sir, I'll have to go back and check it once because like I just wrote it in rough. So that is what I'm asking. Like,
There
is it
are
normal to find
there
it?
are such rare examples, okay, where you can get two different proteins from the same stretch of DNA.
Okay.
Yeah, there are some, but these are exceptions, and as the English language says, exceptions prove the rule. Okay. Yeah, Ankit.
Sir, I have this question. Like you talked in when you were designing these antisense RNA probes, you talked about the cloning plasmid vector. So, like on the two sides of the multiple cloning site, we can have like the recognition sequence for T3 polymerase, and on one we can have the T7 polymerase.
Yeah.
So yeah, so you said if we start from the T3 polymerase, then taking the sense strand as the template, we can have the antisense strand.
Yeah.
So if we reverse this T3 to at the place of T7 and T7 at the place of T3, will there be any difference?
Then you will use T7.
Okay.
So
if we
let me quickly answer something for you.
Yes, sir.
Okay, let me share my screen. Okay, do you see? Can
yes,
you see?
sir. Yes, sir.
You see, there is P blue script KS and P blue script SK. Do you see
Yes, sir.
different
SK plus.
terms? Yeah.
Yes, sir. KS and SK plus.
So do you know what is KS and what is SK?
No, sir.
You have no reason to know. Let me tell you that. So this is very trivial actually. In the multiple cloning site, let me see if this is a. Okay. Okay. Can you see my screen?
Yes, sir.
So you see next to the T3 promoter, there is SAC1.
Yeah.
And on the other end, there is KPN1.
Yes.
So P blue script SK indicates this orientation.
Okay, SK. Okay.
SAC1 to KPN1. But if I go to P blue script KS, you will see the other way. Now, I do not precisely remember what is the reference frame. I hope it is the T3 site only. I cannot be sure. Yeah. So you see, this is now KPN1 to SAC1, and the T3 promoter is still here. Okay. So you can use different variants of the P blue script, or you can change the pair of restriction enzymes. Okay, let me do that another time with you. If you how you clone the gene is entirely up to you, right? So if you put on the 5' end of the ORF, say X bar 1, and on the 3' end of the ORF, say KPN1, then your 5' to 3', so then your gene's 3' end will be towards the T3 promoter, right? But you may want to put the XHO site on the 5' end, and X bar 1 site on the 3' end. Then you can flip the orientation. So these are the planning that you make at the time of cloning your gene. Say you are a very poor lab like mine, and you have only T3 polymerase in your lab stock. Okay, so every time when you are doing the cloning, you will ensure that the 3' end of the ORF must be towards the T3 promoter end.
Okay, the 3' end of the ORF. Okay, yes, sir.
Okay, then only you will always make the antisense with T3 polymerase only. So then you can buy only one polymerase in the lab and you are fine, right?
Sir, and one more thing, in situ hybridization, instead of using the enzymes like alkaline phosphatase, can we use a fluorescence tag also?
Okay, just give me a minute. आप लोग इतना अच्छा अच्छा सवाल करने पे मेरा प्रॉब्लम हो जाता है. You can still see my screen, right?
Yes, yes, sir.
Here. Do you see this?
Yes, sir.
So yes, so this is fluorescent in situ hybridization. Okay,
Yes.
you see how pretty it looks.
Fish.
No, it is
Okay,
fish,
okay,
but
okay.
typically, conventionally, fish is used for DNA hybridization, like when people do chromosomal mapping. This is
Yes,
also
yes.
fluorescent in situ hybridization, but we do not call it fish. We call it RNA in situ hybridization, fluorescent detection.
Okay, okay, sir.
So all you have to do in those cases, the enzyme that will be tagged with the nucleotide will be like peroxidase type of an enzyme. And there are specific substrates on which peroxidase can work and generate blue or green, like green or red fluorescence. Okay.
So there we do not have to use digoxygenin.
No. It will still be DIG, but DIG instead of modified with alkaline phosphatase, it will be modified with something else. Oh no. Sorry. I misspoke. Let me just think through. You do not. Okay, no. You still use deoxygenin only. The DIG-labeled RNA will be recognized by anti-deoxygenin antibody.
Yes, and the secondary antibody will be?
Yeah, the secondary antibody, instead of having alkaline phosphatase tagged, it will be horseradish peroxidase tagged.
Okay.
Yes.
Okay. HRP conjugate, then the substrate will be H2O2.
It is not H2O2, these are like chromogenic substrates on which after peroxidase acts on it, they will generate fluorescence.
Fluorescence.
Yeah.
Okay.
We call them fluorophores. Yeah.
No, sir, like we cannot take like fluorescent secondary antibodies. Like the
that
antibody.
is a very good question. No, that you cannot do. It is not a molecular biology problem. It's actually a sensitivity problem. If you use a fluorescent antibody, like fluorescently tagged antibody directly, then as many these molecules are there, those many antibodies and those many fluorescent tags, right?
Okay, yes.
But if you do a enzymatic reaction, then the fluorescent product is deposited and it is turned over, right? So per enzyme, you might have 100 precipitates, so you get more intense signal.
Okay.
Yeah, it is purely a methodological issue, not molecular biological issue.
Sir, your voice has gone down a little.
Kyun?
Some audio problem.
Is others are also having the same problem?
No, sir, it is all right.
No, sir, it is all right.
No, sir.
From my side.
Ankit, volume badhao. Piyusha, go ahead.
Sir, I had a question regarding application of PCR for tagging of genes by insertion. So the topic was tagging of genes by insertion. So there they were stating that for identifying genes associated with a particular mutant phenotype, a PCR method can be applied. And for that, they were incorporating transposon, but the exact method which they were describing, I didn't got it properly.
Well, I would not go too much into the detail of it, but I will give you the brief idea that how you can tag a gene using fluorescence, using PCR. Fortunately, I have done this technology as well. So. Yeah. Okay. Let's imagine this is a gene, okay? Can you hear me?
Yes, sir.
Unless you respond, I will not know whether you are listening.
Oh, okay, sir. Am I audible?
Yeah.
Oh, yes,
So
sir. Yes.
now, let us imagine that this is the what if part.
Okay, sir.
Okay.
Yes.
Now, if we artificially create a primer, taking from here, okay,
Okay.
outside the WRF,
Yes.
and then so it has been a while for me, but I'm trying. Oh. Then this is a sequence that has that is the tag. Okay, say it is GFP.
Okay, sir.
And Piyusha, I need to think. Huh? Give me 2 minutes to think.
Sure, sir.
Okay, first let me tell you that what actually works. Okay.
Yes, sir.
Then let us try to work backward. Let us say this is the what if, okay?
Yes, sir.
In yeast, the recombination frequency is very, very high. Okay. And I do not recall all the nitty-gritties of the selection, etc. Those are also important here. But I am just giving you the very rough overview of how it is done.
Okay, sir.
It is not the most accurate thing in the world. You will create a DNA construct that will have identical sequence here.
Okay.
Okay.
Okay, sir.
And then in the middle, whatever you put, okay?
Okay.
After recombination, what you will get is, we are talking about the actual chromosome, okay?
Okay, sir.
Like this, okay?
Okay, sir.
Now, here, this green part, if it is the same sequence as the part here, but having a tag, say GFP or something, then you will get in the genome engineered the tagged version of your gene.
Okay.
Okay.
Okay, sir.
But it is not as simple as I showed, because what I am missing, what I am not being able to, okay, like I know how I did it. If you have the misfortune of reading this paper of mine,
Sir, your screen is not visible.
I will share the screen. Just give me a minute.
Sure, sir.
Yeah, if you read this paper of mine,
okay, sir.
I actually have done that. Okay, I tagged one of these genes with something I don't recall now. I think it was with GFP. I tagged it.
Oh, sir, GFP, yes.
Just let me see.
GFP.
Yeah, so I tagged with GFP. If you read it, it is not trivial. Okay, let me tell you how it was done. It made my life nearly impossible. So what was done is that the first round, this green was, this green, imagine that it was a leucine marker. Okay.
Okay, sir.
So if it is a Leu minus strain, then, say I have cloned Leu2 gene here, then my gene of interest will be replaced by Leu2.
Yes.
So it will now be knocked out for my gene of interest, but suddenly the yeast will be able to grow in presence of leucine.
Yes.
Now, in the next step, this same green, imagine that in the next step, the same green, okay?
Yes.
This time it is GFP tagged version of my gene. So the recombination will happen again.
Yes.
It will no longer be Leu positive. It will again be Leu negative. But now my gene will be expressed with GFP tag. So you can simply see the the plates under microscope and see where you see GFP, and you take it and see if it is Leu minus, then you know that it is like you have replaced your gene of interest again, but this time with a GFP tagged version.
Yes.
Okay, so that's how it is.
Sir.
Yeah.
Thank you, sir.
It's only you can do it by PCR. In mammalian cells, it is still difficult. Like mouse knockout is still not possible like that. It is a whole lot more difficult. In yeast, to do a knockout is really 2-day work. Okay. Okay, so I now need to actually take you all through an exercise. Okay. And today, so Nageshwar,
yes, sir.
Please turn on your video.
Yes, sir.
Okay. I am giving you a gene name. Okay.
Okay.
The name of the gene is INT6, capital I, small n, small t6.
Okay.
Organism is mouse.
Yes.
Organism is mouse.
Okay.
Please show me how you will get the, share your screen. I have allowed screen share. Please show me how me and the entire class, how you will get the mRNA sequence for this gene. Everybody please take note, it is very important. There will be questions exactly like this in the exam. देखो मैं क्वेश्चन भी लिख कर रहा हूँ. क्या हुआ नागेश्वर? Internet problem?
Yes, sir. Are you able to see my screen?
Now it has uploaded. Yeah.
Okay. So I'm just going to the NCBI website. So
Try Google. Google is much more powerful. Just write mRNA sequence of this.
So I think it is taking some time. My internet connectivity is slow.
Yeah.
Let me try to turn off my video cam and just keep it in sharing.
I agree. Try
To
it.
see if it is speeding up or not.
Try it.
Yes. It seems to be taking forever.
ये क्या हो गया? You just type in Google mRNA sequence of mouse V6. Who has faster internet? हाँ ठीक है, हाँ ठीक है. Hit it.
Yes. Yes. So I think NCI is totally better. Like from Google, I don't know how to.
Genome browser is faster actually. So like last time you did it with Genome browser. So
yeah, you can try NCBI like UCSC Genome browser as well, but his net speed is very slow. Anyone has faster net speed?
yes.
Try it.
So just a second, I'll share the screen.
Nareshwar, you please stop sharing your screen.
Okay, sir, fine.
So I'll be sharing my screen now. So there is one that I found out, like this is from Genome Browser. Yeah, so I had already selected mouse strain.
Okay.
Now I'll just
put in 6, yeah.
So here, these are the predicted like regions it's showing, like there are exons and all.
हाँ ठीक है, click any one of those.
I'll just click the first one, and here is predicted mRNA.
हाँ ठीक है.
This is the.
Okay. Now you have to clone the ORF into PB script. Okay. How would you do it?
So just a second. So yes, so what were you asking? I couldn't hear.
You have to clone the ORF of this gene in PB
Yes.
script.
Can't hear actually.
You have to clone the ORF of this gene in pBlue script. Transmit. How would you do it?
Sir, can I try?
Sure.
Sir, so if the screen is not visible, so the thing is like we will take the ORF and then we have to add the linkers which will contain the restriction enzyme sites of like any two restriction enzymes we can choose their recognition sequences. Those linkers should contain.
How would you know which restriction sites?
Okay, sir, which restriction sites?
So the restriction sites available in the vector cloning vector we have to choose.
Huh?
We have the cloning vector is
Cloning vector multiple.
No.
We also have to
we have
make
the
sure
cloning.
that the restriction enzyme is also not in the middle. The restriction site is also not in the middle of the.
Yes.
Exactly.
Yes.
So how would you determine that?
Sir, NCBI cutters we have to choose that. What is that like?
Yeah, you go to NEB cutter, Piyusha, you open NEB cutter. Oh no,
I'll stop sharing my screen. Yeah.
No, Akansha, sorry, Akansha, you
Akansha, you are you.
Okay, all right. Yeah, I'll just open again. Just a second.
NEB cutter.
What is it that I have?
NEB cutter. NEB.
NEB, after that cutter, CUTTER.
Sorry, just a second.
It is NEB cutter.
Yes,
New
sir.
Biolabs cutter.
Yeah, this one, right?
Yeah, NEB cutter 2.0. No, the one above that. Take it. This is good.
I think we'll have to visit this.
Now we have to put the sequence here.
Yeah, so go to the other tab.
The mRNA sequence.
But this is the mRNA sequence it's asking for the.
Yeah, so it is fine. It is actually shown as DNA sequence. You go from ATG to the stop codon, all the capital letters.
ATG.
ATG, from the ATG to the. Yeah.
Control C, Control V, yeah.
Yes.
Now just do the digestion. Submit.
Submit.
Okay, now there should be a list of say zero cutters. You click on the zero cutters, list zero cutters. On the right hand side, ha. So these are the enzymes that do not cut, right?
Okay.
Now you open another tab.
Then select plasmid vectors.
Now open the map for P-blue script.
So P-blue script.
Yeah.
So should I do the add gene?
Snap.
Just open any one of these. Just click on any one of the images, you will be able to see the image. Go a little below.
This is the, yeah.
Yeah, so here.
So is it visible?
Yeah, now you get to see, yeah, close that stupid thing. Now you get to see all the multiple cloning site enzymes, right?
Yes, sir.
Now check with the zero cutter list, which are the multiple cloning site enzymes that are not there, that are there in the zero cutter list. So I can see KPN1. No, on the other tab,
Yeah, just a second. So ACC 651 also.
Yes, the first one is there.
In P-blue script?
Yes, sir. So this one.
Okay.
Yeah.
Then KPN1 is also there.
Let me see ACC 651 sequence, restriction recognition sequence in the ND cutter. Okay. Yeah, this is the same thing as ECOR1.
Okay.
GGA TCC, na?
GGA.
It is either ECOR1 or BAM-H1. You do your stuff, let me quickly figure out.
It is BAM-H1, sir.
Yeah, it is GGA TCC. It is the same thing. So you now write down somewhere that BAM-H1 is one side, and the other one you choose again, go down the list.
So there's KPN1 also.
Yeah, there is KPN1, there are a lot of those. I see NAR1, I see, so you take KPN1, so you now write down BAM-H1 and KPN1, okay?
Yes, sir.
Now you go back to your P-blue script map. This is a bad idea because BAM-H1 and KPN1 sites are right.
They are the same.
They're right next to each other. That's not a good idea. So try to find something a little far away. At least one of the enzymes should be, okay, so I see BAM-H1 here. Okay, so BAM-H1, KPN1, so can you go back to the zero cutter list again? BAM-H1 is not there, but this SEC 651, I don't understand that part. Why that is the case.
Is BAM-H1 GG8
Acha, this is
GGT is.
So there is a slight difference. Okay. So you see BGL2. You see BGL2 on P-blue script. I'm sure BGL2 is there.
BGL2.
This is BPG. I'll just enlarge it a bit. Oh. Sorry.
Sir, BGL2 is not there.
अरे ये इतना उछल कूद क्यों करवा रहे हैं?
नहीं सर, वो like the cursor.
अच्छा, fine.
PGL2
Then
PGL2 is not there.
so search for another enzyme. Plow1. Let me see.
So SAC,
one is
SAC2 is there, I guess. SAC2.
Yeah. So now you have KPN1 and SAC2, right?
Yes, sir.
So now tell me what are you going to do? Now show me how you will design the primer. You go back to the RefSec, like to the actual RNA-seq. Yeah, SCC 651 SAC1 can also be used, Nageshwar, you are right. Now you tell me the primer sequences. Type it out.
So
We can go for the primer designing tool on NCBI.
गोली मार देंगे, अपने आप से करो, देख के.
Sir, so the 5 means to 3 prime would be this ATG.
Yeah, the 5
Here
prime
will be the same. Yes.
The forward
It will be
primer
the same
Sir,
as
will be same, but we have to add the restrictions site of KPN1.
Okay. So Akanksha, do this.
Yes,
Copy from ATG
Yes, sir.
to this CTC. Na, utna nahi.
Okay, okay. Sorry, sir. Sorry, sir.
Like much less. Typically, you want to go 20 nucleotides.
20 nucleotides.
20.
Okay, fine. Control C.
Yeah.
Open a Word document. We will come to your Word document later. You don't have to show us the Word document right now.
Yes, sir.
Control V.
Yes, sir.
Okay, now go to the bottom of the gene, end of the gene.
Yes, sir.
So copy the last 20 nucleotides, including the stop codon.
This much should be fine.
I cannot see the screen, but that's fine. Again, put it on your Word document.
Yes, sir.
Now share the Word document. Now stop share and reshare the Word document.
Yes, sir. Is it visible?
It is visible, right? So now you put the restriction site sequence for the first one. The first one was
KPN1.
KPN1.
So you, I will tell you the KPN1 sequence. You don't have to jump around.
Yes.
Okay. It is GGT ACC.
Yes, sir.
Show me where you will put it.
The GGTACC. Okay, this is the primer.
Give another color.
Akansha, you can put another color for that.
Yeah.
Very good. Okay. Now design the 3' primer, the reverse primer. What you have is the last few nucleotide sequence, right?
Yes, we have to make the complementary.
Complementary.
Write 3 and 5 in the appropriate places, please. Let me see.
So this is the 5' end. The 5' end. And I copied it from that side. So that will be, this is 3'. And this is 5'.
Yeah, that's the one I was talking about. Now design the reverse primer.
So it will be T.
Type.
Okay, sir.
You will have to do all of this in 2 hours.
Yes. T.
Na. You like you always type on the before, like type T.
Yeah.
No. Where you were typing, you were fine. Go there where you were typing.
Yes, sir.
Type T. Use the sidebar to come before T again.
Okay, all right.
C.
Yes, C.
Again, keep on doing that.
C A. Will it be a T after this, right? T. G.
TCA.
ठीक है चलेगा for illustration purpose चलेगा. Put the 5 prime 3 prime here. Very good. Now put the SAC2. I will tell you the SAC2 sequence. It is CCG CGG.
CCG
CGG.
It's a reverse.
Reverse, I think.
Reverse.
I am not able to follow actually.
Why?
So like this, okay, 5 dash.
You have
So
to.
this will come here, like
actually.
No, location is fine, but the sequence should read from the 5 prime end
Yes.
CGG.
So it will be from 5 dash. It will be CC GCGG, right?
Yeah.
Yes.
Okay, yeah, that's CC G. CCG.
Arey Baba, the same way I taught you earlier. No.
Yes, no, sir. This is what.
Delete.
No,
First erase it, and then
delete
write.
everything.
Yes, please again.
Type C.
Yeah, C, and then yeah.
Yes.
Again C. G. C. GG. So now this is your reverse primer. Okay.
Yes.
So now when you will PCR amplify it, if you put your reverse primer, you put it in say green color, then you will have in 6 sequence. On one end you will have the red colored KPN one side, and on the 3 prime side you will have the green colored SAC2 side. Okay.
Yes.
Then you will digest it with KPN1 and SAC1, and you will digest your P-Blue script with KPN1 and SAC1, mix them together, add ligase, and then transform it and get your bacteria, possibly with blue-white selection, to get the cloned ones, right?
Yes, sir.
Yes, sir.
Now, Akanksha, you go back to your P-Blue script screen, like stop sharing and reshare your internet wala screen.
Yes, sir. Just
Yeah. Now, let me see who else is there in the class today. Okay. Anupam Mishra, are you there?
Yes, sir.
You explain to me how blue-white selection works. क्या हुआ सो गए?
I don't remember actually. I'll study.
So
Nothing
can
to
I try?
No. There are lots of students in the class who are simply trying to hide. Ashish Kumar,
Yes, sir.
explain how blue-white selection works.
Sir, actually, this method works alpha.
Huh? Hello? आशीष का क्या हो गया? No, not you. Muktesh,
Hello.
Yes, sir.
try it.
Sir, here it is, which is the lag Z, that is the gene. It codes for the beta galactosidase enzyme.
0. You will get 0. What is written there inside that arrow, solid arrow?
Lag Z A
Alpha.
alpha.
Okay, now that is a hint I gave you.
So whenever any gene is not cloned into that, that enzyme will be functional. So that when when it is transformed into a bacteria, and that bacteria is grown in presence of a X-gal, this enzyme will convert that X-gal into a blue product, so that the colony will appear
This
blue in
is
color.
not the whole enzyme. That is why I will give you 0. मैं बहुत खड़ूस हूँ ये सब मामले में. Okay, I see hands raised. Okay, Piyusha, try it.
Sir, I did not raise my hand. It was from previous, I guess. But
Okay, now fine. Shreya, you want to try it?
Yes, sir.
Okay, go ahead.
Sir, so this beta galactosidase enzyme, it is basically composed of two separate polypeptide chain, the alpha and the omega.
0.
So,
That is not correct. Who can give me the right answer? Ayush, I will come to your question little later. I see a lot of raised hands.
Raising.
Nagesh,
I am raising hand.
Priyanshu bolo. I've never heard Priyanshu speak.
Hello, good evening, sir.
Yeah.
Sir, I haven't been able to catch up all the lectures, but I recall something from my previous learning. So this LAGZ alpha and the other portion of this enzyme is encoded by the genome. So they both combine to give a functional beta galactosidase. So when the colony is grown in presence of X-gal, the beta galactosidase can cleave the X-gal and give the blue colonies. But when we have cloned a gene in the LAGZ alpha region, and it causes the insertional inactivation of this LAGZ alpha gene. So the LAGZ alpha is not able to complement with the other like part of the and is not able to form the complete enzyme. Therefore, the colonies will appear white.
Very good. I will give you 8 out of 10. So Shreya, this is not, beta-galactosidase is not made of two peptides. Beta-galactosidase is a single polypeptide protein enzyme. It is an artifactual discovery that if the peptide is broken into two parts, so that one has the sequence of what is shown here corresponding to LAC-Z alpha, and the other part is LAC-Z omega, these two parts of the same polypeptide can come together and complement and give the full enzymatic activity. That's why it is called alpha complementation or intramolecular complementation. Okay?
Yes, sir.
Priyanshu, right?
Yes, sir.
Okay. Priyanshu, you tell me that will this work in any E. coli strain?
Only with those E. coli cells which express the other portion of this enzyme.
Absolutely correct. Okay. So, you can write down and check later the genotype. Okay, why don't I do this? Akanksha, please take down your screen share. Can you see my net? Okay. You can see my net, right?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. See the genotype here. Can you see here?
Yes, sir.
RK minus, ND minus, HSD R17. These are all features that allow, like restriction, digestion, etc. to be turned off. And I do not remember all the ones, but here is the one that you are interested in. This part. Okay. This is a constructed bacterial strain where the omega part of the LAC-Z is in the genome.
So only the omega part or the alpha part is also there?
No, only the omega part is in the genome.
Okay.
It is basically a LAC-Z minus strain.
Okay.
Where the
Yes,
omega
LAC-Z alpha minus strain.
No, LAC-Z minus strain. Here, even this little part, like the LAC-Z delta M15, this part has been inserted using bacteriophage. Okay, that is rather complicated genetics. I do not really remember the whole thing. See, here it says that LAC-IQ-Z delta M15 gene on the F prime episome. So episome means like it is artificially inserted. Okay, so therefore the blue-white selection only works on bacteria that has this kind of genotype. There is other very popularly used E. coli strain. I'm forgetting the name now. Where you will not be able to do it. Okay. So this is the secret of blue-white selection.
Yes, sir.
Sir, actually from here many questions are coming. So the first question is like, if we take a strain which is producing the fully functional LAC-Z,
then it will not work.
Okay,
then even without plasmid, it will give you
the blue color.
Blue color, yeah.
Okay.
Yes.
Sir, so we cannot do this blue-white colony selection with bacterial strains like BL21 or
no, BL21
DH5 alpha,
is the name
right?
I was forgetting. BL21, you will not be able to do it. BL21, since we are onto the genotype, let me alert you to the special property of BL21 DE3. I love this strain. I have spent months and years on this strain.
It is a good strain to express recombinant
Yeah.
proteins and
Yeah.
bacteria.
So see this D3 part, it is a lysogen that encodes the T7 RNA polymerase. T7 is a bacteriophage, right? T7 is a bacteriophage RNA polymerase, which is not an endogenous gene of E. coli. But here it is a genetically modified E. coli, where T7 RNA polymerase expression is under the control of lac operator. Okay.
Okay,
So,
sir.
in your expression system, when you induce with IPTG, you basically induce the expression of T7 RNA polymerase. And it is a two-component system. The plasmid used is PET5A. Here you see that it has the T7 promoter and the ribosome binding site. Do you see that?
Yes, sir.
So
Yes, sir.
the T7 RNA polymerase is being induced in the bacterial genome. Engineered genome, all right, but in the bacterial genome. That will then bind to this T7 promoter here and do the transcription in this direction. Within the transcript will be the ribosome binding site, and that will lead to the translation. Okay, so these are the final points of cloning system, expression system, etc. And
Yes, sir. One more follow-up question. Like, if we forget about this alpha and omega thing, if we have the whole LAGZ sequence, okay, and it is disrupted by the insertional inactivation, then we have to like transform it into an E. coli, which is LAGZ negative background.
Yes,
Na, sir?
yes, yes.
Okay.
The only problem there is the LAGZ sequence is so large that the vector will be unwieldy. Here it is
That is why.
accidental discovery and people took full advantage of it.
Okay.
Okay. And you know, there are these TA cloning vector. What they have there is what you were trying to say.
The whole LAGZ is there.
No, it is TA cloning vector does not use LAGZ. It uses a bacterial toxin. I am trying to. बुढ़ा हो गया हूँ ना, सब याद नहीं रहता है. Yeah, here.
Is it some CCD toxin?
CCDB.
Yes.
Okay. So the CCDB gene, the whole gene is inserted like they are in the multiple cloning site. Okay. So this CCD, so these are modified versions. Forget about it. This is the CCDB. So if the CCDB is uninterrupted, then that bacteria that is transformed with the plasmid where cloning has not happened, that bacteria cannot survive.
Yes, sir.
Only if the CCTB is interrupted, then only the bacteria will survive, and then there is antibiotic selection. So because of this, TA cloning vector is very, very powerful.
So when we do a toxin selection, why do we again need an antibiotic selection in that?
You need the antibiotic selection because of the untransformed bacteria so that they do not grow. Okay, like your transformation efficiency is at max 5 to 7%.
Yes,
so the remaining 95% will grow.
Yes.
Okay. I think Ayush had some question.
Yes, sir.
Yeah, Ayush, go ahead.
So I have
Typed
written in
it.
chat box as well. Yes. So my question is, since we are using KPN1 as a linker in the forward primer, and it recognizes GGTACC where it is cutting one nucleotide at the 3'
Ayush,
end.
when you are cloning in
Hanji.
P-blue script, expression of the protein is not your objective. You are only cloning the gene. Like we will come to expression later. Then we will
Okay,
use
sir. Okay.
highway.
Sir, and one more thing, we have used this CCG, this SAC2 CCG, but when we have, like I have experienced sometimes, we have GC clamps, and GC clamps should not be there in primers. So, like we have to put again some new nucleotides to.
Yeah. So it is actually not like that. Nowadays, as Akanksha or someone suggested, like MIT has a primer designing software, so that can help you design the primer. But it is not so simple if you provide the precise thing. Like if you have no option but to start from the ATG and end at the UGA, then the primer design software is not particularly useful. Akanksha, can you put up your screen another time, the Word document?
Yes, sir. Yes, sir. Just a second.
Yeah, I have exactly 10 more minutes.
Yes.
So what we used to do, Akanksha, can you quickly count the number of G and C's in the, like first you highlight the whole sequence, the top, the forward primer, and see, like delete the 5' and 3' part. Highlight it.
Yes, sir.
So go to the letter count.
So 10 words.
No.
Oh, words. Okay, yes. Letter count.
So 23, right? So you delete some the last 6. Close it. Delete the last 6. Take it. Now delete this 5' that is written there.
So with the restriction enzyme.
No, just the 5' part.
Okay.
Now count how many G's and how many C's are there.
So in the entire sequence.
In the entire sequence, how many G's and C's are there? इतना कामचोर होते हैं आज के बच्चे.
But only in the forward
14.
primer.
Only
Sir, 14, sir.
No, only in the forward primer we have to calculate. So 6 G's and
14,
sir, 14.
Okay, and what are the total number of nucleotides?
So 23 minus.
I think it will be around 23.
23, sir.
Yeah. 24.
24.
Oh no, 23.
23, right?
No
So
space.
if there are 14 GCs, then 9 ATs.
No, sir, we have to calculate only for the forward primer. No? In forward.
In the forward primer, if there are 14 GCs, then there are 9 ATs, right?
Yes, sir.
Yes.
So in our time, we used to do a very rough calculation of G into the G plus C multiplied by 4 and T plus A multiplied by 2.
Yes, sir.
Okay, so 56 plus 18. So 74 would be the rough TM of the forward primer.
Yes.
I do not know what people do today.
Sir, 74. Like the reaction, I mean, like the extension is happening at 72 degrees. Right?
It does not
So,
actually.
like,
It does not actually exactly work like that. Okay, I will tell you what we used to do. The नीचे का length भी तो थोड़ा गड़बड़ है. What we used to do, and even today what you must do is, I will not test you for that in your exam, but in the lab what you have to do is you have to ensure that the TM of both the primers are close to each other. Okay.
Yes, sir.
Otherwise, the temperature at which the forward primer will anneal, the reverse primer will not anneal or will anneal non-specifically, and vice versa. Therefore, that's where what Ankit was talking about. Then you have to add nucleotides here and there to kind of balance the TM of both the primers.
Okay.
But you do that within the inside of the gene, not in the overhang part. Okay, what is in red, we call it overhang because that is, you do not have complementarity of that on the template, right?
Yes.
So you don't care about the overhang. For the interior part, you vary the length of the primer to kind of get the same kind of TM in both the ones. Okay.
Okay, sir.
That's how it is done. So now you know exactly how to clone a gene, right?
Sir,
Yes, sir.
with the textual knowledge, we know that a primer length should be 18 to 22. But
Yeah.
like in according to the laboratory methods, what is the permissive length?
We typically go inside 16 to 18 nucleotides.
Okay, okay.
Like if you leave the red part aside, inside we go 16 to 18 nucleotides.
Okay.
Fine?
Yes, sir.
So then we have to discuss microarray because there will be questions on microarray. So do you want to do it tomorrow or day after?
Sir, tomorrow because
Yeah,
Saturday's exam.
So that you have a little bit time to internalize it, right?
Yes, sir. Tomorrow.
Tomorrow 8 p.m. again, right?
Okay.
Yes, sir.
Okay, send me a WhatsApp message around 6:30 to remind me.
Okay,
Okay, sir.
Okay, sir. Chalo, bye.
Bye, sir.
Bye, sir.
Bye, sir.
Bye-bye. Good night.